Catabolism is one of the two major divisions of metabolism. It represents the set of biochemical pathways responsible for breaking down complex molecules into simpler ones. Through these degradation reactions, organisms release the energy stored within chemical bonds, making it available for cellular work and survival. Catabolism consists of metabolic pathways that break down complex molecules into simpler ones. Think of it as the body's way of dismantling large, energy-rich compounds. During this breakdown process, the energy that was stored in chemical bonds gets released. This released energy is then captured and used to power various cellular functions throughout the body. Catabolic reactions have four key characteristics. First, they are exergonic, meaning they release energy in the form of heat and adenosine triphosphate, or ATP. Second, they are degradative, breaking complex structures into simpler components. Third, they are oxidative, involving oxidation reactions where electrons are removed. Fourth, they are convergent, meaning multiple different pathways converge to produce common intermediate molecules. There are four major catabolic pathways in human metabolism. Glycolysis breaks down glucose into pyruvate. The citric acid cycle, also called the Krebs cycle, converts acetyl coenzyme A into carbon dioxide and energy carriers. Beta-oxidation degrades fatty acids into acetyl coenzyme A molecules. Finally, protein catabolism breaks down proteins into amino acids, which can then be further processed for energy or converted into metabolic intermediates. The energy release mechanism in catabolism follows a specific sequence. It begins with the oxidation of carbon-hydrogen bonds in nutrients. During oxidation, electrons are transferred to carrier molecules called NAD+ and FAD. This creates the reduced forms NADH and FADH2. These electron carriers then deliver their electrons to the electron transport chain, where the energy is ultimately used to synthesize ATP, the universal energy currency of cells. Catabolism employs four main types of degradation reactions. Hydrolysis uses water to cleave bonds, breaking polymers into monomers. Oxidation removes electrons from substrates, converting carbon-hydrogen bonds into carbon-oxygen double bonds. Decarboxylation releases carbon dioxide from carboxyl groups. Phosphorylation transfers phosphate groups from ATP to substrates, activating them for further breakdown. Each reaction type serves a specific purpose in the overall degradation process. Catabolic pathways are tightly regulated based on the cell's energy needs. Catabolism is activated when ATP levels are low and MP levels are high, signaling energy demand. The hormone glucagon also activates catabolism during fasting states. Conversely, catabolism is inhibited when ATP and NADH levels are high, indicating sufficient energy. The hormone insulin inhibits catabolism during fed states when nutrients are abundant, and the body shifts toward energy storage. Let's review the key points about catabolism. First, catabolism breaks down complex molecules into simpler ones, providing building blocks and intermediates. Second, it releases energy through oxidation reactions that transfer electrons to carrier molecules. Third, major catabolic pathways include glycolysis, the citric acid cycle, and beta-oxidation of fatty acids. Fourth, catabolism is carefully regulated by the cell's energy status and hormonal signals to maintain metabolic balance.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.